Thank you for joining our second NCBIB webinar, and the second one is on the data science to innovation, from data science to innovation. And my name is Behruz Javisari. I'm director of the National Technology Centers. With me, Chi Duan, program director, and Andrew Witz, also program director, in helping in this presentation. And then all the PI that it will be introduced at the time of the presentation. My goal of the webinar is basically, my objective is to learn about the P41 Center and their mission and their tools and technology, and then also to promote the community awareness, because there's a lot of people may not know about this, except those that they close to the centers. And also, I like to encourage collaboration among the technology centers to be able to collaborate together. When I'm presenting, I'm going to show that there's an overlap on the science and the area of the disease, that they can help each other. And also, I like to promote the collaboration with the community and the service project and the training. In the first webinar, actually, was that they already doing a joint workshop on the overlapping training. So it will have four center here that it will be introduced later, and then each one has 20 minutes to talk, and we keeping the question hour for 30 minutes in the end. Please keep in mind that if you have a question, write in online, ask, mention it, who is the question for. It would be easier for me or my colleagues to relate the question to the person. So it's good to just say, okay, this is for Dr. Toga, and then, or even if it's for me. Thank you. I'm going to start with a brief overview of Biomedical Technology Center. Their foundation is a 3 or 4 technology research and development projects. These are as I said, the foundation is the research and technology project. These are distinct projects. These are multidisciplinary, but they are interact and they are interrelated, and ambitious effect leading the practical tools. Sorry. In top of that, there are collaborative project and service project. The collaborative projects, the projects that they are from the active funded project. It doesn't have to be agency funded. It could be industry funded. It could be just a university funded, as long as there's a funding for the project and is active. They have a push-pull interaction. It means the center technologies get pushed these collaborative projects, and they take advantage of this for the advancement. And then in response, they push the center back to additional new technology development. Again, this is not consider optimizing the existing technology, is basically the pushback. It means that, okay, a new development for the technology. And then the service project is the same way, that the center pushes the technology to the service, and they take advantage of the service, and they do not push back, or the center does not pull from the service projects. They can, after a while, they can become a collaborative project, or the collaborative project can become a service project. In top of that, there's a training that the centers offer training in the area of that they have technology and method that they develop in, and they offer the training for the broad range. They offer it for the student, postdoc, engineers, scientists, and clinicians. They also do a dissemination of what they develop through the publication, through the licenses, and through the patent. In top of all of this, there's administration that oversees that within the university that oversees the project. I'm going to get to a little more detail. They are technology research and development projects. They have to have a synergy, and there's the administration in the top, then there's a training and dissemination. But if you look at this, you will see that each TR&D has SP that is a one-way and a CP that both-way, but one TR&D can work with the multiple CP or SP. As I mentioned, they are technology, multidisciplinary. They should have a national impact. They have to be very unique. When I'm talking unique, is not only the applicant, is not only the location, it is the technology. Main point is the technology uniqueness. What they offer is unique. When a new application comes in, my first question is that, what is unique? Why should I come all the way to Boston to get this? I can just go to the Johns Hopkins next door. So it has to be something unique about that. They have to be innovative, complex but distinct, and they should be high-risk test-bed and ambitious effort for leading the practical tools. They have to be looking for translational application. So I mentioned it, they should be driven by CP and SPs, and then they should have a training and dissemination that includes patent, public outreach, website, and presentation. And they also should have a administrative, that these are the senior project, that the PI or the lead PI, they have to have a funding and funding history, and they have to be in a senior and pioneer in their field. This is the centers we currently have. We have about 25 centers. They're distributed in a different area. That we have MR related centers, and then there's a optical imaging and machine learning. There's a different area that we have about 11 different technology that centers support. The modalities are not the technology. I like this. These are the logo for the different centers, and this is that they cover all the electromagnetic wave spectrum. And you see that the same logos exist in different places. It means there is an overlap on their area that they work on. And that's the people that I look for to come to the webinar, the one that they overlap with each other, that they can take advantage of each other's service. And the same thing with the biological cells. There's an overlap between different centers. Some center, they are working 3 areas or 4 areas, and other ones in some other one. The impact is great, and this shows the impact, the CPs of the active application through the United States, and there's about 271 collaborative project currently, and then there is this one shows the CP and SP, 271 CP and 240 SP on the active projects. Is also have, they work internationally, so the collaboration and service can be international. And some other things that the centers, they do not only support the NIBIB, they support all other centers. In here, there's about 13 or 14 centers that they, ICs that they take advantage of. And the NIBIB has the most number. But there's a next NCI, another one that they take advantage of the service. There's also international collaboration. There's also NSF and other agency will collaborate as well. They do have a good publication record comparing with the other area, like with the R01, there's much better. This is old data. I just asked last week for the new one. We have about 4,000 publication combined from the centers, active centers, but the citation is about 120,000. So it's much better than what I have in here. Thank you. This was my talk. So I'm going to ask Chi to introduce. Oh, I forgot to mention, if for those people, they don't know about it, if they're looking for anything on the NCBIB, the National Technology Center, if they just type NCBIB and NIBIB or just NCBIB and my name, they will get to this page. There's all the information about it. Is there information for the grantees on the how to write the progress, the template for the white paper and the other one. And you guys, I can always, if there's any question, contact me. Okay, I'm gonna stop sharing and she can introduce our next speaker.
Hi, thanks, Beirut. This is Chi, and for the next one, we have Dr. Toga and Thompson for the to present the Center Laboratory of Neuroimaging Resources. Dr. Toga, the floor is yours.
Thank you. So let me just begin by saying it's a privilege to be able to do this, so I appreciate the opportunity. Thank you all. But it's a bit bittersweet. We are in our sunset year, so our resource will be shutting down at the end of this fiscal year for us. And it's a sad day. We have a lot of love in our hearts for working on this. We've been alive for 26 years now, and I think we've been enormously productive. And I'd like to show you a few slides that describe what our center is about, and also to tell you a little bit about our productivity. So let me just go through this slide very, very quickly here. So we have modeling computational solutions for a variety of data types, include imaging, genetics, behavioral, and clinical data. There are 3 TR&Ds and has all the components that were just outlined as requirements for the P41. We've been in business for a number of years. For the last, I don't know, 5 or 6 or 7 years. We've been, I'm sorry, for 9 years, we've been over here at USC. We moved in 2013. Collectively and across the entire period of our 26 years, we've produced almost 4,200 peer-reviewed manuscripts, abstracts, books, etc. Trained almost 560 visiting scientists, postdoctoral scholars, graduate students, and undergraduate students. We've had about 160 training events, and the projects that have been associated with our center are more than 635 and include 251 collaborators with about 437 service projects. We have a variety of infrastructure and facilities that are made available to our collaborators and service recipients. Much of that takes place in a building that we have here on the USC Medical Campus. But of course, all this is electronic and is distributed in a variety of ways. We serve up a great deal of data. We currently have about 16 petabytes of data that are in use as part of these projects, with availability of 22 petabytes. We host, manage, and process over 9 billion files and have roughly 45 million new files added every single month. These are the 3 TR&Ds. I'll get to the data science one in a minute. The second one, which Paul will talk about, is diffusion MRI and connectomics. And the third one is intrinsic surface mapping, which Yonggang Shi will describe to you. In addition to those activities, we of course have the training and dissemination activities that are required for a resource. And we provide training for all manner of students, from undergraduates, as I mentioned, to postdoctoral fellows and visiting scientists. Different training mechanisms enable us to do that, from demo days, educational activities, and so forth. And that's led by Meredith Brasky here, who has put together a variety of workshops and seminars over the years, some of which are listed here. There are also formal training programs. We have a master's program that's associated with our center, which has turned out a number of students who have gone on to industry as well as academic careers, which has been a great boost to their careers as well as enhanced our center. And we have associations with underrepresented groups, such as this project with Cal State Fullerton. So we've extended this beyond to other institutions that serve underrepresented groups. The TR&D one, which is data science, is described here. This is run by Scott New and myself. We develop and combine a variety of software components needed for scientific data management. We've created an environment where analyses are coupled to the data repositories and the archives, and data from research studies can be QC'd, discovered, reused, and I'll show you some examples of that. The structure of that is illustrated here, where we have a variety of cohorts coming in from projects that are either ongoing or have already completed. And then analyses can take place on those, results from the analyses coupled back to the original source data. This is a slide from the IDA, which is the main data science repository. There are 140 active studies in the IDA presently, with serving active 141,000 users. And in the data center itself, there are 82,000 subjects which have genetics, imaging data, behavioral data, clinical data, as I mentioned. These data move in and out from 161 different countries. Now, this won't activate right now because I'm not in presentation mode, but if you get on the website, which is open, you will see all of the distribution of where the data goes and where it comes from. If you look at the activity here, this is not live, but there's somewhere around 260 million total downloads and somewhere in the order of 1.8 million uploads, serving, as I mentioned, about 143,000 users currently. The data systems that are provided here allow a variety of search. Interestingly, a lot of the projects are longitudinal, and therefore temporal search design had to be developed more recently, which is shown here. We've also allowed people to search using graphical interfaces so that you can find a cohort that answers your particular question by moving these sliders to restrict the cohort limits by age or scores on tests or other factors. And then you can link all these together so that you can have very specific cohorts that are identified by limiting the values in any number of variables. All of that is interactive, online, and connected to the data systems. During this last year, we've added a number of projects. You can see some of these listed here, and we cover a variety of neurological disease, psychiatric disorders, and others, again, with all of the data modalities that I mentioned before. Genetics presents some unique challenges. So we have lots of data. We have both the CSV file, a variety of SNP files, but we also have the BAM files for those people that want to take the raw data that's not yet aligned and can do things with it. And so in this particular one, we have the SNPs from 23andMe. There's an API integrated into the database, and you can do all kinds of things online with that. We've also added a variety of new analytic capabilities. This one is coupled with data from Parkinson's disease and the Michael J. Fox Foundation, and it allows us to do online analytics on the data that's contained within the data systems. So, which I went too fast here, sorry. So the number of studies has grown every single year for the past number of years. And we now allow people to do searches and manipulate data from across studies, which increases the power tremendously, allowing people to aggregate data that comes from different studies when the permissions are granted. And we support, as noted before, there's about 140,000 active accounts, but these are the number of investigators for which receive considerable support. There are a number of other activities in the data science, such as motion correction, QC, AI algorithms, where there are connected GPUs where people can process the data online on our systems as well. And the final slide that I wanted to show you is an interactive augmented reality system that allows us to look at these data in an augmented way. This is now being shown as part of the abstracts and presentations for the upcoming OHBM, Organization for Human Brain Mapping, and I think it's IPME, one of the other ones, has also adopted this. So it's been accepted by a number of journals as well, and we think this is yet another way to interact with complex data sets. So I've used up a little bit of time here. I'm going to turn it over to Paul to talk about TRND2. So I stop share, right?
All right. Thanks so much, Arthur. Can you see this slide okay? Does this look okay?
Yep.
Okay, great. So I think, as Arthur said, the P41 resources consist of technical research and development projects. This is the second of three, which is co-led by Neda Jahanshad here at USC. And you'll see many of the algorithm developers' names are listed there at the bottom. This has to do with diffusion MRI and connectomics. So we have a lot of collaborators who are using diffusion imaging to look at fiber integrity of the brain. They want to see factors, disease, development, genetics that affect the microstructure of the brain. They want to analyze fiber tracts and bundles. I'll tell you a little bit about a toolbox by Ryan Kabin that's been used by many, many collaborators looking at frontotemporal dementia, brain injury, HIV, special cases of tractography in infants. And then also they want to look at brain networks and connectivity. So we'll talk a little about some tools and algorithms we developed that allow something called discriminative fusion of brain networks. And we'll go into a little bit about the novel mathematics that involves. So just on the neuroscience side, we supported the largest ever diffusion MRI studies of the brain in 10 diseases. I won't list them all. You'll see in red there a couple of the collaborators who provided unique data on HIV and neurogenetic syndromes that are actually pretty rarely studied with diffusion MRI. And just to tell you about what is done with the data, if we have better models of diffusion and also brain microstructure, we can get at some of these disease effects with more sensitivity. So we'll talk about one in particular that enhances a measure that's widely used, fractional anisotropy, which is computed usually from the diffusion tensor. But this is incorrect in areas of the brain where fibers are crossing. So we'll talk about some methods we developed to fix that. So here's, I borrowed this slide to look at diffusion in the brain when there's a fiber there or when there's two fibers there. We need models of these diffusion processes that are sensitive to the anomalies that go on in disease. You can see how complex this is. And unless you get the fiber directions correct, other techniques that sit on this, such as tractography and network analysis, will also go wrong. So just looking at this in detail, here's a DTI reconstruction. And if you look in more detail, you can see the full complexity of this. So we've been writing tools that essentially measure what is going on in each voxel using either continuous mixtures of tensors, which you can see there, or all built up to model the multiple characteristics going on in the tissue, and measures that agree with microstructural measures that are commonly used, but add a little more sensitivity and signal to noise in these very complex fiber crossing situations. So just practically, I mean, if you were a collaborator with data on Alzheimer's disease and you came out with the results in the top row, you wouldn't be very pleased. I mean, that would be the standard method. The assessments of the differences in white matter microstructure between people with dementia and controls, and also associations with clinical dementia rating. If you use the enhance measure, this is a tensor distribution function measure of FA at the bottom, it's much more in line with what people understand about the topography of dementia effects on the brain. So this has been used widely. It's been used to study links with brain microstructure and cognition, also in longitudinal studies that you see here that show that if you're looking at microstructure longitudinally, these measures offer much better signal to noise. Now, a lot of these tools are in a toolkit. So Ryan Cabine, who's junior faculty member here at the INI, put together this fabulous toolkit called QIT, Quantitative Imaging Toolkit. And you can see the link to download it on the wiki here. And he put together a terrific number of workshops, courses, even a dedicated program as part of our master's program to teach students this. If you're a remote user, you can set it up. There's an installation page. There's data sets, in fact, HCP that you can download and experiment with, and a guided set of videos that Ryan Cabine has put together on how you would load this up. Now, this is terrifically enjoyable to work with. It's not only a good viewer for all brain data, as you can see here, but it takes you through visualizing microstructure, how to make tractograms, how to parcellate these if you want to manually edit them, or if you want to use AI. He has this terrific set of essentially wiki pages and videos that take you through these methods. So I would just encourage you to check Ryan Cabine's QIT wiki out to look at that. Now, on the more mathematical side, diffusion data varies from scanner to scanner and from protocol to protocol. And Dan Moyer, who is a student in the lab now, he's faculty at Vanderbilt, developed this incredible method to mitigate the cross-protocol differences, realizing that if you run a machine learning algorithm on data from a certain protocol, it may fail rather badly if it's applied to data from another protocol. He uses deep learning methods, variational autoencoders, and adversarial methods that essentially reconstruct data to look like it was collected with a different protocol or with a different scanner. So you can imagine you're trying to merge data from many, many sites. You can even enhance or modify the data so that it looks like a particular type of protocol. Now, this was evaluated, a very exciting challenge at Mackay, where you can assess whether or not you've done a good job of harmonizing data by using ground truth data from people that have been scanned with multiple protocols. Another line of work, we've done a lot of mathematical tutorials on how you use AI, adversarial models for harmonization of diffusion data, other things. You're welcome to take a look at some of those. And then finally, last 2 minutes, improved methods for connectomics. So many of you are very familiar with the quest to understand the connectivity of the brain, usually based on a parcellation and a tractography method, and some system to quantify the matrix of connections. One of the approaches is that these methods often struggle in data from people with dementia or low quality data. You can see on the top right there that different tractography methods might actually extract different things, which would give you a very different perspective of what's going on. So this is a method that runs 9 different tractography methods to, in this case, discriminate dementia from healthy subjects. One of the ways that you can leverage having all these methods is do a machine learning task of learning the best convex linear combination of networks that arise from these to classify a disease. You can imagine this is a discriminative fusion approach where you put all of these methods together and fuse them and see which combination is best for a given task. And actually, in evaluations versus the discriminative fusion of the top method here, which try and detect a certain disease, they vastly outperform all of the other methods run singly. So this is rather a nice boost for people who are interested in connectomics. This is going into really cool directions. There's work using deep learning. Many of you are very interested, as I know, in AI, embedding essentially generative models of connectomes and using these in different contexts for neuroscience-related questions. So before I hand over to Yonggang, about 30 peer-reviewed papers a year coming from this project, including the largest studies of 10 brain diseases, mainly focusing on improved microstructure models, a nice tractometry toolkit, which I encourage you to take a look at, some very cool math to harmonize diffusion MRI, and then finally adaptive fusion to study disease effects on connectivity, and there's a list of methods there. So Yonggang, I will hand over the relay baton to you, and I think you will have the last, 5 or 6 remaining minutes. So over to you, Yonggang.
Okay, I'll try to be fast. So you can see my screen, right?
Yep, looks good.
Yeah. So I just tried to highlight some of the project we did in TIND3. So basically, the focus of TIND3 is on shape analysis algorithms. So mainly we have developed algorithms focusing on using some mathematical tool called the Laplace spectrometry spectrums on manifolds. So instead of conventional people using canonical space for parameterization surface, which is such as like a spherical mappings, all right, for surface registration. So we use some tool called this is an eigenfunction of Laplace spectrometry operator on the surface, and then using eigenfunctions as a coordinates for high-dimensional embedding space. The nice property of this embedding space is basically it's isometric embedding, and if two surfaces, they are isometric, they automatically have the same embedding, and we can get it in their correspondence or mapping easily. But certainly, different surfaces have different shapes, different geometry, and their embedding will be different. And which is exactly one of the reasons we don't like to use spherical registry mappings, and it's because we have different shapes between anatomical surfaces and a sphere, and because of different shape, you're gonna have a lot of distortions, right? So what we propose is like we gonna embed the surfaces into using the eigenfunctions of Laplace spectrum operator, and to try to find a map in the high-dimensional embedding space by optimizing a set of metrics on the surfaces to make sure their embedding will be very similar, and that's you can eliminate some of the unnecessary distortion just because of a parameterization, right? This has been published a few papers previously, and so this method, I will not go into the mathematical details, but they are kind of general. We don't make too much assumption about surfaces. We have applied them to cortical surfaces and various subcortical structures. And here we just illustrate some examples on mapping like subfields of hippocampal surfaces as a validation of the techniques. So on the left, you can see like a demonstration examples like source and target surfaces, and the subfield labels were generated by the high resolution T2 images from free surfer tools. So what we did with surface mapping is we only use the T1 weighted MRI, and using the mapping algorithms, you can pull back the labels from the target to the source, as shown on the bottom row. You can see that compared with the spherical registration, you can compare the pullback label with the source labels and see that the Amos algorithm produces a better agreement. And you can see on the right side more quantitative results of the dice coefficients of those subfields on the hippocampus, and compare with the results with the spherical parameterization-based registration algorithms. You can, generally speaking, our method achieves much better results on a relatively large data set from adenine. So, and also because these methods are general and they are not just usable for closed surfaces, we can also apply them to surface patches. So you can do more dedicated mapping, focusing on like a specialized anatomy in different anatomical regions. Here we demonstrate a surface patch in around like entorhinal cortex, because it's an important area in the early detection of brain pathology and atrophy in Alzheimer's disease. So on both the left and right, we show the detection of pathological tau signals in the entorhinal cortex, and also the brain atrophies, which shows that compared with like a surface spherical mapping based on pre-surfer, our method can be more effective in detecting these early changes of brain pathology and brain atrophy in the entorhinal cortex. So not only we do like these surface mapping algorithms, we also generalize these tools to do more recently to do vasculature modeling in some of the novel applications. One example we show here is something called OCT angiography for detecting or visualization and analyzing microvascular changes in the retina, right? So this is probably a few papers listed here. We are one of the first groups in doing a 3D analysis of this OCT angiography. Typically in the field, they only do like a 2D analysis of retinal vasculatures. And also more recently, we're generalizing these methods to do like a cerebral small vessel diseases, collaborating with our colleague in the Institute, Dr. Wang. And so also, we also apply develop some novel geometric models to enhance fiber tracking algorithms. It's kind of in synergy with like a TIND2 by Paul. So we are applying a novel parameterization with smooth curves called parallel transport frames. And this leads to a new fiber tractography algorithm. Not only you can detect, reconstruct smooth curves, but also can incorporate some of the property called topographic irregularity, which is common for many of the motor sensory tracts. Here we show also example to the preservation of retinotopy in the reconstruction optical radiation. We did quantitative validations with some of the retinotopy information from the visual pathway, right? So to summarize, and we have been doing some of the cutting-edge development of a surface mapping algorithm based on Laplace spectrometry spectrums. They're applicable for both very different kind of surfaces. Right? We also generalize into modeling the microvasculatures in brain and the retina, and also develop some of the novel fiber tracking algorithms with the PTF-based parameterization. And the tools are publicly available as like a tracker software, developed by my postdoc Baron Edelgen. So that's it.
Thank you to our three presenters there. So I'm just going to move us along because we're a few minutes behind schedule, but just a reminder, if you're in the audience, please, if you have any questions, put them in the Q&A, and we will get to those after our next couple presentations. So I'd like to introduce the next presenter, Dr. David Kennedy, who is a professor of psychiatry at the University of Massachusetts Medical School. And David and his team are going to be presenting on ReproNIM, which is a Center for Reproducible Neuroimaging Computation. Take it away, Dave.
Thanks a bunch. Can you hear me and see me, I hope?
Yes,
thumbs up on
your
that.
slide looks good. Yeah.
Great. Okay, and I drew this short straw when we created this project, so I present stuff for all sorts of other people. So much of this work is done by many others. We'll name them at the end, but it's a great pleasure to be part of this P41 program and to present with these other esteemed colleagues and present our information on the reproducibility issue. My 20 minutes of fame here will quickly remind ourselves that reproducibility is a problem. I hope I don't have to spend that much time, but we'll remind ourselves of that. We'll talk about what this ReproNIM structure is that is part of this P41. We'll remind ourselves that part of the reproducibility crisis comes from the fact that everything we do in these complex imaging studies matters in terms of the interpretation of the results we generate. And we'll look at some potential solutions in terms of the more re-executability in our publications and a sort of a framework to support that re-executability. You don't have to look very far in the literature to see lots of doom and naysayers about where science is, cluster failures, and much of what we know is wrong, and the problem with fMRI. I'm not going to dwell on these topics. They'll be in the slides I share, and you can read those and weep at your own discretion. But it's not just imaging. Science as a whole gets lots of occasional bad press about too much trusting and not enough verifying, issues related to assuming that software just runs, and the problems that come out of these types of assumptions. So again, lots of issues that point to these kinds of things. Then, of course, the whole statistical world gets upset about various things. Most published research findings are false, and other doom and gloom types of articles come out. So again, it's not too hard to get sort of overwhelmed with how much problems and issues emerge in the field. And again, you have to look to editors of the Lancet worried about the scientific literature being false. Scientists sculpt data to fit their preferred theory, significance, and are sort of hanging on the importance of a particular point p-value is really causing problems. So all that said, the core problem that I come down to is in our applications of trying to use these lovely imaging and analytic technologies to really make important inroads into various mental health and neurologic types of issues. And so while the literature is has lots of observations, it led Dr. Kapoor, Phillips, and Insel in 2012 to really lament that there's a profusion of these statistically significant findings, but they end up being sort of minimally differentiating and hard to to turn into actionable types of things. And our way of publishing those make it such that it's hard to properly replicate these, and we end up looking at findings in ways that neither can be confirmed nor refuted. And so that's, yes, good for getting grants, it's good for getting publications, but it's not so good for solving critical issues in medicine. So that's kind of the background impetus for ReproNIM, a Center for Reproducible Neuroimaging Computation. Its goal is to help improve the landscape of reproducibility in neuroimaging in order to extend the value of our large investment in neuroimaging research. It has the 3 TR&Ds that were prescribed. The first one is on software and data discovery. Second is related to the proper implementation of description of metadata and workflow and provenance descriptions. And the third TR&D is to really implement execution environments that take advantage of the tools that monitor provenance and metadata, but do it in a sort of computationally efficient way so that it scales to both local and sort of global computations that use local and globally shared data. And of course, on top of this, there's a big mission to provide training and education to the community to use these tools and evolve the way they're thinking about doing their neuroimaging observations in a more reproducible way. This kind of summary slide here kind of reminds us in the old days, and anything I complain about, I'm guilty of. So take all of the naysaying I do about the current state of the art with that grain of salt. But traditionally we acquire data, we run analyses, do computations on it, we store that data locally for a while, we write our publications, and then we publish that, and hopefully we then share that data that went with that. That's good, but what we're missing is a lot of the how this data, was generated. So for every computational and analytic step, there's provenance and metadata that's also being generated. So what in the past we've not done a great job of monitoring, having our software tools or our data tools monitor their own metadata, and each transformation we do doesn't necessarily monitor precisely the provenance of each of those transformations. What we're doing is adding, this metadata and provenance tracking functionalities, so that when you do your study and then go ahead and publish it, you can publish, yes, the data, but also the metadata and provenance information that goes along with it, so that when you see a published observation, you can track that published observation to the particular values, to the results of that, back to the data from which it came, and the sort of provenance track for how that data was generated. So it expands our current sharing infrastructures to really get the how and why different elements get generated in that context. So going back to our sort of high level problem, approximate replications can rarely be refuted nor confirmed. Well, my conjecture here, it's impossible to confirm or refute something you can't first replicate. And then in general, we do not publish things with the necessary details in order to replicate. And when it comes to generating a re-executable observation, it turns out that everything you do in that processing stream matters. Not to belabor these too much, you can look at the slides and the publications that are in, but exactly what operating system you're running on matters. Exactly how much data you use matters. Exactly which tool you select matters. Exactly what statistical model you use to analyze the data with matters. And each of these things can make things that replicate or don't replicate, or extend or don't extend when used on the same and different data sets. So that sort of sets up this little sort of spectrum of reproducibility that we like to think about. When we go off and publish an article, we have a bunch of data and we have a particular analysis approach we take, and we generate a particular result. Execution is really can someone other than the author, or even the author later, find that exact same data and perform that exact same analysis and generate that exact same result. And to some extent, you want to be able to do that in order to then more systematically explore the generalization of that original finding. So by systematically explore, we want to take that exact same data, but then be able to manipulate exactly how that analysis is done. If we do a similar analysis, we should get a similar result. Similarly, we want to lock that data down, or lock the analysis down, and use similar data or expand our data set, and we should generate a similar type of result. And in general, a good, robust finding should, be able to take nominally similar data and a nominally similar analysis and generate, a similar type of result. So again, we see this attempts, sort of to be done, but when we don't publish our methods exactly, it gets too easy to get a different result and not know why, different results appear. So we need to be able to understand that when we don't replicate something, what is the source of that non-replication? And again, that really is cornerstone of that is the ability to do, the re-executable version of a given publication. So if the cornerstone of reproducible research, therefore, is transparency, transparency is achieved through annotating and sharing precisely what operations were performed and on what data in a fashion that could potentially be re-executed by someone in an attempt to replicate and ultimately document the generalizability of that original finding. So that leads us to the concept of what a more re-executable publication of the future would include. So of course we have to have the lovely words that humans read and understand and give us the sense of what was going on. But in addition to those words, we need access to the actual data that went into that particular observation. Now again, access to the data doesn't necessarily have to be to everyone, but someone with appropriate authorization ought to be able to get to that same data. We need the workflow, the analysis workflow, to be specified precisely so that someone else can run that exact same analytic set of operations. We need that execution environment to be locked in and documented properly so someone else can get to that exact same execution environment. And we need that complete set of results that go into then the subsequent statistical analysis to be available so that one, the transparency of that can be determined, and two, that if someone wants to replicate a finding or re-execute a finding, they can verify that they're getting the right answer at that particular stage. So in summary, we want a completely re-executable research requires this complete description of the provenance and the metadata of each of these different elements. Now, one could imagine, fancier, publications, but even with current, publication methodologies and supplemental, data and supplemental, linkages to, different elements, it's not super, rocket science to be able to take a publication and connect it to the data, that goes in, to it, and workflow specifications, that can be available at GitHub, or execution environments that can be incorporated into Docker or Singularity, and results that can be shared at different the data repositories and things like that. So that again, in principle, it's not really rocket science to create re-executable publications, but it just takes the interest and concern and a little bit of time to devote to doing that type of thing. So that prior publication is sort of a trivial something that could be re-executed by others. A much more interesting type of re-executable publication comes in the context of trying to replicate some of the historical findings from the literature. In this case, if you look at a classical figure from Ganjavi et al. Using the NIH pediatric database and looking at brain volume and corpus callosum area, you get a lovely PDF and you get lovely pictures, and you can read the words of what data was used and what software was used. So the re-executable version of that you can generate on that same data set, the similar types of analyses and similar types of observation. But in a re-executable version of that, each point in this graph knows who the subject and the demographics of that subject was, what MRI scan generated that particular observation, what scanner, what acquisition details, and where one can find that data source. They know what workflow generated that, what software, what hardware, what parameters. And you can know where that observation fits into the sort of semantics of observations of different types of things. So specifically, the workflow that generated it, can be, embedded in a Docker or an image, that can then be re-executed by others. So in order, to try to, accomplish, doing, this sort of systematically, we really need to try to, embrace a more comprehensive set of functionalities when we perform our imaging experiments. And we need to try to plan everything in advance. We need to do version control of everything, data, scripts, software, containers, protocols, all need to be under version control so that when you come to the publication, you know exactly which set of these tools and operations were accomplished. Everything needs to be annotated. In this case, we want sort of semantic annotation and the description of where measurements fit together, and that's both for the raw data, the derived results, the processing, all are necessary so that different tools and different observations can be semantically linked back together. And we need to be doing this using standards, so that others can access our data. And when it comes to image processing, standards can be through the uses of containers, Docker, Singularity types of things, and everything needs to be published and shared in some way so that others, future you, future people in your lab, or people who want to access and re-execute and extend or generalize your observation needs to be able to access the data. Someone needs to be able to access the data, the derived results, the workflows, etc. So overall, thinking of these sort of phases of the research process, from design, collection, and processing, statistics, and publishing, and these sort of best practices of planning, version control, annotation, standards, and publications, there's a lot of tools and resources that can be developed and implemented by investigators to try to fill in these different elements. And so ReproNIM is working on getting these types of tools into the hands of the end users, making them easy enough to use. We don't want to change the way you do your analysis drastically, but we want to make the ability to do things with these intentions much easier to accomplish. So again, so trying to get functionality sort of across the whole workflow that supports standards, and getting data standards and semantically represented, and using containers for your applications, and expressing the results of your analyses in sort of semantically complete ways, and doing your statistics in semantically complete ways, and being able to publish and share not only the results but the semantics and the metadata of those results as a whole. So doing this all, is hard. There's a lot of tools and practices, that can be invoked, and we're trying to work on and promoting those types of things. So what is it that we want, the end user, to think about? Well, there's just a lot of things we need to learn. And so we support all sorts of training things I'll show you a little bit more about. As an individual, we're just trying to get people to be more mindful of each of these steps and take more control over the transparency of your ongoing process. As a center, we'd like the center support their investigators in using more of these functionalities that increase the reproducibility of their research outputs. And then we want to see yourself as a software developer or yourself as a data user in the constellation of resources that ReproNIM is helping to support, and to add your tools and resources to the support of these types of semantic and metadata and provenance tracking. There's a bunch of training resources, including modules for fair data, computational basics, reproducible workflows, and statistical tools. There's a training fellowship that has trained over 60 trainees to FAR, and there's sort of a train the trainer mechanism. So we encourage people to look into that. We've been involved with a number of R25 ongoing courses, one with ABCD and one with Repro Rehab, so to try to create the workforce of more reproducibly aware investigators, and then monthly webinars and lots of videos and tutorials of that sort. Encouraging centers to adopt for their users a more comprehensive set of tools that invoke training and version control and containerization and management of metadata and provenance, and lots of many tools that we interact with, trying to make sure they become more provenance aware and make it easier for their users to track the provenance and metadata as they go along, so that it's easy for the end user to use any of the different tools and accomplish this tracking. I'm not going to read them here; they'll be available. But there are a set of 10 collaborative projects that we're working with, and again, we're supposed encourage more folks who have push and pull types of topics to interact with us. We're happy to work out different interactions, and there's a large number of current service projects from a number of different areas that are trying to use those collaborations to push our technologies out into the broader community. And again, we encourage folks to work with us and extend this framework for your use and for your tools. And again, there's many people who are part of this, and we're happy to talk at further length on any of these particular topics. And this is who runs our various elements. And thank you for your attention.
Yeah, thank you. I do see, given the time constraint, we'll move on. I do see we have a few questions, and just a reminder that because we have multiple presenters today, it would be helpful if you can specify which one you want the question to be addressed to. We will have the Q&A section later. So for the next, we will have Dr. Carl Frederick Westing to present the Neuroimaging Analysis Center. Thank you.
All right, so I'm going to talk about our Neuroimaging Analysis Center, NIC. And see here, I first showed the team. And so the leadership is myself and Ron Kikinis. We have 3 TRDs. One is led by Paulina Golan at MIT, and one is led by Sandy Wells, and I had one core. And the training and dissemination is led by Sonia Pujil and Mike Halley, and Steve Piper is kind of in all cores. I'm going to as a translational aim. So we actually also already at 25 years, so time flies when you have fun. So unfortunately, we are sunsetting now this year. It's been a nice five cycle of this grant. And kind of the main theme has been all the time is kind of understanding the human brain through imaging and how to use this for advancing the role of neuroimaging in healthcare. And one driving kind of thing for us has been that, I mean, it's been actually very exciting. Every year, our imaging devices become more and more powerful. improves faster images, high resolution, and even more new modalities. So this is really like an opportunity to kind of do help healthcare within in vivo measurements. And the focus of the center has been finding new ways to extracting kind of disease characteristics from advanced imaging, and then also to kind of give out our method as tools that can be used as open source and in collaborations. Before I continue, I just wanted to say a few things about kind of what we kind of the overall what we do. I mean, when you say image analysis or medical imaging, it's kind of a little bit vague, and different people think differently about this concept. And kind of what has emerged now is something we call medical image computing, and it's actually a term that now is taking up, like MICA is a conference that had this term. There's the MICA is medical image computing. And can you go to, if you're interested, go to Wikipedia. There's an entry that actually Ron Kikinets initiated a while back, and we have all been trying to help to kind of describe what we mean. And it's really more than just an image analysis. It's an intersection by different kind of big things like fields like computer science, information engineering, electrical engineering, physics, mathematics, and medicine. And you need all these kind of components to be able to do something that's actually relevant for helping to find kind of measures for different diseases. The second part in your imaging analysis center is, of course, imaging. And we found that many of you others, we have heard earlier today, that even if you have the best image analysis tools and the best team, sometimes you cannot get the information that you need because it's simply not there. So we kind of wanted to work on actually, when you kind of hit those roadblocks, go back and see if you can change the imaging to get more information. So it's kind of interplay, how you do imaging and how you do the analysis. So they're really together, and that's been a theme in our center throughout the years. So we have 3 TRDs, and I'm not going to go into too much detail because as we stand sunsetting, we will not continue to work exactly in this. But one TRD that I'm leading together with Yogesh Rathi is about kind of new diffusion MRI. We call it multidimensional dimensional MRI. So like finding new microstructure models for tissue and also sequence development. The second TRD that is led by Paulina Golan is statistical inference for imaging in disease. And one part is kind of image imputation. I'm going to say a little bit about later, and statistical inference of disease. And the third one is led by Stanley Wells and Lauren O'Donnell, and that's image features for brain phenotypes. And they work a lot with tractography and large data sets, and try to find clinical disease pattern in these large data sets. One thing that's been kind of a theme throughout these 25 years, but also now, as you see in all the TRDs, is machine learning. And as you all know, deep learning has become very kind of popular recently, but as you also know, machine learning has been around for a long time. And in the early days of NIC, we were doing mostly statistical machine learning, like supervised learning, and then also clustering, and then later manifold learning became popular to reduce number of dimensions in the data sets. And now more and more machine learning is kind of synonymous with deep learning. But when we work with machine learning, we kind of try to use all of these components, and deep learning is of course very powerful, but it's not the only way to do machine learning. Steve Piper has an aim in each of these TRDs, and that's been helping very well with the collaboration between the cores and TRDs, and also how to translate the software and clinical validation. So it has been a good experience to have this kind of glue aim, through the last 2 cycles of this grant. So I'm not going to go through too much of all the technology in each TRD, and partly it's because we're sunsetting now. But and this is also for you guys that don't have P41. A very large part of these grants are really training and dissemination of the the science that is kind of and technology is developed. And seminars has always been a good way to communicate, but also tutorials and other lectures and dissemination events have been very kind of powerful. And especially now with after COVID, we kind of had to retrain a little bit how we do. We cannot just go out and teach in different conferences. So Zoom has become a more important part, and it's actually been opening up so we can reach more people. And now even if we have events where we go to meetings, we try to have hybrid meetings. Has been very kind of a good way to open up to more participants. And many of these activities is enabled by our software platform, the 3D slicer that's been developed through these 25 years. So, as I mentioned, the dissemination events, we have had basically since the start 2 meetings per year. It started with one in the summer at MIT, and then we had in the winter in Salt Lake City. And after a while, we changed around a little bit to different places. And right now we have one in the summer at MIT, or had that for a while, and then in the winter we have had in Las Palmas. And the reason we moved to Las Palmas is that then we could reach much more of the European researchers. They don't have to go as far, and also of course it's warmer than being in Boston, and it's been very successful. So last year we had this then was virtual because COVID was still going. 170 participants from 20 countries. And it was exciting that 41% of these were first-time attendees. So kind of a very kind of energetic kind of meeting. This year we can actually have the meeting in person. In total, we had 249 people there, virtually or in person, and that was from 28 countries. So it's really amazing the reach of these project weeks. And even higher first-time attendees at 52%. And you can see here a quick summary of the different countries. So we are mostly from US, but also many people from Europe, Spain, Germany is kind of the biggest countries. We also have a lot of participation from Africa, Senegal, and Mauritania, for example. And that is because we have collaboration project, and they have very easy access to kind of go to Las Palmas. It's very short. So we have new interesting collaborations with African countries. This is actually a little bit a new example for a new trend. We talked about collaboration project and service projects. And we normally had service project was more users using our tools, and the collaboration was kind of push-pull, as Perus mentioned, that we kind of were working together to create a good tool. This is actually more like a dissemination project, but it's more like on steroids, because these are not users, these are developers that took Slicer and they looked at the tutorials we had, and they created now a plugin using the Monai label, which is kind of from this medical open network. So Monai, it's a large open source platform for deep learning, and Monai label is a way to help annotate. So I put some links down there for what happened in the project week, but also linked to Monai there. So over 100 participants, and this is an example of kind of dissemination that we didn't think about in the beginning that could happen. And we're really excited when people just find our software and take it to the next level and kind of let us know later. And of course, we are in the loop now, but a lot of this happening even without our help. We also do the traditional web dissemination. We go to conferences, and RSNA's kind of been a big one that Sonia Pujil have been going to every year. And last year we released version 5 of Slicer, so that was a kind of the theme last year. And also we actually reached 1 million downloads of Slicer last year in February. So we were very excited about that. And this version 5 has some new visualization tools to show 2D, 3D, and 4D MRIs and CT data, and also PET, ultrasound, nuclear medicine. And also has some new interesting segmentation tools and editors. So another thing that happened also during the past year that was exciting, we got a grant from the Zuckerberg Foundation to supplement some of the work we're doing in NIC. And that was to add new languages to the tool. And to begin with, we had to use kind of the engineering team. You can see some of them here up to the right, left. And you have Steve Piper there and Sonja Pujil from NAC. And what happened in the beginning, we had to change the infrastructure to be able to add different languages. And after that, the people from Senegal and Mauritania, together with their engineers, started to put French language in the slicer. And of course, it doesn't hurt that Sonja Pujil is native French speaker. And then after that, in the same project, we're also working with Mexico and Brazil to add Spanish and Portuguese. So that's been an interesting support. And now in the background also, this is more grassroots work. We also have, you can see lower left, maybe it's hard to see, but it's both Chinese and Japanese is happening now. So it's exciting now, and now we'll be able to reach even more people with these adding new languages. So that's an interesting and exciting addition. Here's some statistics. I said we reached 1 million last year in February. I download this today, so it's 1.2 million downloads, a bit more. And it's about 500 daily downloads, and it's pretty impressive, I think, and I'm really excited about that. And you can see the different regions, and mostly North America, but also Eastern Asia is also very close to the same kind of downloads. If you look in countries, you have the same order: United States first, then China, then Germany, Italy. But you have even a lot of downloads from Turkey and Mexico and other people you wouldn't first think about, but I mean, we're excited to work with them. So that was software. And I also wanted to maybe plug a little bit about the research impact. And now we're looking at publications, and these are only the one, I mean, not the only, but the ones that actually has PubMed index, so that's how we search for them. So we have here 867 papers and 2,509 authors. And thank Mike Halle for creating this. And what was also interesting is not only how many papers, we wanted to figure out a little bit what the impact was, like the chain effects. So if you take out the top row, so these papers resulted in 62,000 citation in 45,000 a bit more papers from, you can see a lot of 128,000 authors. And then that in turn resulted in 1.5 million citations. So that's one way to just see how your work kind of would impact. Another thing that was also interesting to see that this semantic scholar, if you looked at, they actually have something they call highly influential citations. So they have some machine learning to figure out what makes a paper highly influential, and you can read more there if you go to this website. But apparently 4,000, this NAC publication influenced 4,000 papers strongly, which in then turn had 14,000 influential papers. So it's a different way to see the impact, and it seems we would like to learn more how to read this, but I mean, I think we're excited about the numbers. So I'm going to say a few things about the research. I'm not going to go in too much detail, but this is just a map of what we mentioned before. We have 10 collaboration projects and 10 service projects. And of course, I cannot go through all of this. I will focus to go through a few examples where we have collaboration with other P41 centers, because that's a little bit the theme of today's webinar. So I will only, and I only picked local P41 centers because I think also the idea here, if I understand it correctly, is try to figure out how we can continue to work together more between the centers. So, so this is one collaboration that Yogesh Ratihar, he's in TRD1 and with Kevin Sepapop at the time when he was in Boston. So this is Bruce Rosen P41. This is how to generate very high resolution DTI images or diffusion images. And with this kind of 0.66 millimeter resolution, you can really see much more detail in the U fibers when you do tractography. And this is virtually impossible if you have a standard 2 by 2 millimeters. That's in the lower right there. So if you really want to look at the cortex and the structure, the microstructure there, we need high resolution. So this has been a nice collaboration. Lauren O'Donnell in TRD3, collaborating with Claire Tempeni's P41, that's the NCBIB we're going to hear about soon. So this is working with Alex Kolbe, the neurosurgeon, and looking at anatomy and also how that association tracts and language are affected. This is collaboration between Paulina, and Bruce Rosen's P41, sorry, and so with the Eugenio Iglesias. And this is actually quite important work because we talked a lot about deep learning, and some of those techniques we see in the literature and out there, they're very powerful when they've been trained on one types of data, and you can do amazing segmentations. But if you change even the scanner or just change some parameters in your protocol, some of those methods completely are thrown off. So they are not robust in the way we would like them to be. And this project here was kind of designed to do the opposite. How can you really robustify? We give data sets with different size voxels, different contrast, different orientations, and see if we can generate high resolution data from that. And even if you have a T2, can generate a T1. So, and this is not really for looking at, for diagnostic purposes, is to get robust data out from the system, because we need that for registration. We need it for many tools we have, we'll take it for advantage. So getting this robustness into deep learning is very important. And once we have that, we can continue to go back and do this. And we heard earlier about normalization, different scanners. This robustness is really important, and this is work in this direction. The last thing I want to talk about technology here is something I've been working with the team here in Boston and Lund. We call it multidimensional diffusion MRI. And the reason we're trying to look at new contrast is that, for example, if you have two very different tissue types, and they're kind of schematically shown there under, you have one box there, we can call it cells that are all the same size, but they have different orientation. So there is a variation or variance in that signal, but has nothing to do with size, just orientation. And on the other side, on the right, there is a variation that is only in size, so completely different microstructure. But if we measure, we kind of standard techniques to diffusion, the measurements look the same. So on the vertical axis, you have the signal strength, and the horizontal axis, you have kind of the B value of the gradient strength encoding. And this is, of course, not what you want if you cannot distinguish this kind of easy changes in cell size or cell's orientation. But by adding more advanced diffusion encoding here, the colors mean different direction in gradients, so actually changing the diffusion gradient encoding during the measurement. And then on the left side, you get the straight line. So it looks like only one type of cell. And on the right, you can see that it's the same to have variation. So this is like to get another dimension to see differences we cannot see before. So adding dimensions increases the specificity. And this connects to my last slide here. So to get even better diffusion MRI and microstructure imaging, we need scanners with stronger gradients and stronger slew rates, so how fast you can ramp up. And we got a grant recently from Mass Life Science Center, and this system called Magnus. It's a gradient insert that you put in a 3T system from GE. And with that, we can get 300 millitesla per meter, which is the same as the Connectome scanner now that Martino Center, and 750 Tesla per meter it's very high slew rate, better than the Connectome scanner. And this kind of machine more compared to the Connectome 2 scanner that's going to be up and working soon at Martino Center, which has slightly higher gradient strength, but maybe slightly lower slew rate. So they're going to be very comparable, and ours will also have an upgrade path. So this is very exciting, and end of this year we'll have 5 systems running in the US. Tom Fuji has one already, and we're going to have Walter Reed, University of Iowa, University of Wisconsin, and one at Brigham. And there are 2 more that will happen next year. I cannot say the name, it's not official yet, but we're going to try to build a consortium with these systems. And this could be a very nice way to collaborate with different P41s, because we have seen many of you are working on this kind of high performance neuroimaging. So we're planning to work with all the P41s that fits this, and I'm also, anyone wants to work with us on that, please contact me. So I think I stopped there in interest of time.
Yeah, thank you. For the time being, let's move on. And for next, we have Dr. Claire Tempany and her team to present the center titled Advanced Technology National Center for Image-Guided Therapy. Thank you.
Thank you very much, Chi and Behrooz, for this opportunity. Can you hear me okay and see my screen? I'm moving to presentation mode.
Yes, everything is good.
Everything's good. All right, great. So many thanks for this opportunity to share with you an update from the AT-NCIGT, which is the Advanced Technologies National Center for Image-Guided Therapy. This is a new P41. We're in the beginning of the 3rd year of the first cycle of this, continuing to do the same work as proposed initially. We have 3 TRDs, and the training and dissemination core, the same structure as everybody else. The names of our TR&Ds and the people are all on this slide, but I'm going to go through approximately 9 short projects to show you the breadth and the exciting work that is going on in each of these TR&Ds. So I'll introduce people as we go through it. First up is the TR&D 1, which is imaging of cancer heterogeneity. This is led by Natalie Agar from the Department of Neurosurgery at the Brigham. And the aims of this group are to develop and validate new technologies to characterize tissue in vivo and ex vivo at both microscopic and macroscopic scale. And more specifically, that's really taking 2 modalities, one being dedicated mass spectrometry imaging and MRI to provide these probes of tissue metabolism and microstructure. And one of there are 3 aims in this group, and the first aim is focused at looking at MSI, or mass spectrometry imaging, and the deliverables include sensitive imaging of metabolites using a MALDI 15 Tesla device, photographed down here, with the MALDI-TIMS-TOF method for multi-scale data integration and coding implementation for data analysis. Much of this work comes from our strong relationship in the Department of Neurosurgery in the evaluation of brain tumors, which has been a legacy program from the prior P41 and even before that. So today, Natalie and her colleagues do in vivo mass spectrometry imaging on specimens from, ex vivo on specimens from brain tumor procedures performed by Dr. Golby and her team. And this is just one illustration of a recent publication that demonstrated very nicely how the oncometabolite 2-HG alters T cell metabolism to impair CD8 T cell function. And I'm no expert on this, but I will attempt to interpret some of this for you. And looking at this image here, you will see the 2-HG in color purple, and then the T cells, the CD8 cells in green. And you can see that these have been impaired in many of these areas. And so it is a surrogate marker between in vivo imaging and final pathology, but mass spectrometry can provide this exciting new metabolic assessment on these brain tumors and allow us to understand them better. And another illustration in the whole space of brain imaging and brain mapping is the integration of multiple different sources of information. So this slide illustrates the concept of using EEG mapping with the subdural grid placement here, integrated into a brain MRI image with gadolinium enhancement of various areas of tumor and treatment. And you can see the grid template overlaid. And then the same areas spatially localized to allow for mass spectrometry data to be acquired in the exact same places. So allowing for very direct three-dimensional localization and correlation. One of the collaborations that Dr. Agar and her team are working on closely with the Mayo Clinic with Jan Zakaria is CP1, and this is an illustration of some of the data from that clinical trial, which was looking at tumor, infiltrative tumor, normal brain, and necrosis. And we can see the various parameters of the MR sequences that correlate with the various pathological findings. And so the depth of the multiplexing of both imaging and other variables has become really possible through this great work. And one of the things that's really important, of course, in this correlation business is to ensure that we have continuous monitoring during the removal of the brain tissue, and we know where everything is coming from in three-dimensional space. And Sarah Friskin leads this effort along with Alex and many others on the team, where we're tracking the devices and the surgical instruments during a tumor resection to map this resection cavity. And one of the important issues here is really to determine the presence or absence of residual tumor. And so you can see the concepts on these slides and a publication that recently resulted from this in an effort to determine what's happening when we resect, when Alex is resecting a tumor here. You can see residual tumor, false negatives, false true negatives, and areas where we think there is tumor left behind. And this is really a closed loop iterative process, starting with the preoperative imaging, model generation, correlated into the surgical view, which is directly seen here, and then using the brain lab and other techniques, we're using the navigational views also. And one of the key features that Sarah has provided is the ability now to do intraoperative ultrasound imaging fused with the brain MRI, as you'll learn more about in the next couple of minutes. But Sandy Wells's team looking at registration and segmentation overlays of the data so that they're matching. And so this provides updated information all the way through the surgical procedure. Also in TRD1, I mentioned that we're focused on MRI, and in MRI and AIM2, led by Stefan Meyer, we're looking at new approaches using diffusion MRI to assess prostate cancer. As you may or may not know, diffusion MRI pulse sequences have radically altered and really enhanced prostate cancer MRI techniques, providing an in vivo marker of the detection for the detection of clinically significant prostate cancer. And this has led to the explosion of prostate cancer MRI across the world, really. And this is every day getting better and better, but we really need to go deeper into the diffusion techniques to understand what can we learn from this and what more can we learn about the microstructure. And as CF mentioned in NAC, the multidimensional MRI data has been worked on between himself and the group in Lund and Stefan Meyer as well. One of the things that's critically important with diffusion is to ensure reproducibility. As we also heard from David Kennedy earlier, reproducibility and reliability of the data across platforms, across vendors, really challenging with diffusion imaging in prostate. And so one of the solutions that Stefan has been working on, which is published just this month, actually, in MagRes Medicine, is a thing called truly reproducible uniform estimation of ADC. And so true ADC is a nice concept that will allow us to really look at different vendors, different models and reproducibility retrospectively. It's done within the same scan time with full protocol flexibility. And this graph is demonstrating the plot of the true ADC here. It's a fitted curve, and each of these orange dots represents one of 20 B values that are acquired in this sequence, comparing it to the clinical acquisition, which is essentially usually 3 B values, 0, 800, and something like 1400. And you can see illustratively on the right side how they're very comparable resulting images between the clinical and this new form of imaging using the true ADC approach. And this will really help us, I think, ultimately provide fitted DWI images also here on this slide, again illustrating similarity to the clinical approach, certainly very effective and very reproducible, we hope. So what, in summary, this is going to allow us to calculate ADC independent of B value. And I didn't define ADC as the apparent diffusion coefficient, the metric that's extracted from diffusion imaging. And this is done in the same scan time as a clinical protocol and can be computed for any B value. And so this, I think, is very exciting and will allow us to do a lot more multi-center trials and quantification of ADC going forward. The third aim of TRD1 is led by Junichi Takuda with help from both Stefan and Natalie in the effort to really map tumor hypoxia. As many of you will know, if there's hypoxic areas within a given tumor, it becomes very radio resistant, for example. So it's not so easy to treat the hypoxic regions with the same dose of radiation therapy as you would use for the non-hypoxic or the normoxics or even the hyperoxic areas of a tumor. So there's heterogeneity within a given tumor. So we're learning to try to map this in vivo without having to use Eppendorf catheters or other invasive ways to measure oxygenation within the tissue. And so we're looking at a time series of T1 and T2 mapping and data processing software to allow us to understand this better. And one really interesting a way of doing this is being developed by Junichi and a team of people from MIT and Strategen Biogen, a company. We can see that they have developed this little silicone elastomer oil mix, which is put into the catheters that can be placed in vivo in the body during a radiation delivery procedure. And this type of procedure can be a brachytherapy procedure where the catheter will be inserted with the little silicone tip bead inside it. And this will allow, and this is an overview of a poster recently presented, but these silicone can be imaged and will allow for quantification of oxygen level within the silicone, reflecting the local tissue environment. And this has really been proven possible in the first 7 people, and we're going on now to do further studies of this and make this more reproducible and reliable as well. As part of a collaboration with Strategen Bio and the Department of Radiation Oncology here at the Brigham, Junichi and his team have been awarded an STTR and another one pending to work through further development of validation, development of an improved sensor, and then test this in an animal model. And so that is the ongoing status of that particular project and all of TRD1. I'm moving now on to TRD2, which is led by Sandy Wells, who is working in the area of deep learning to try to analyze and make these novel imaging tools available for image-guided therapy through state-of-the-art algorithmic approaches. And the first aim is looking at modality agnostic representation using 2 new methods, which is learning via hierarchical variations and autoencoders. And this is the team here. And this is done as one of our CPs with Tom at King's College in London. In summary, that what they've been working on is to really use this new approach of representation learning and focus on both the finding compact informative representations that will really allow you to be modality agnostic when it comes to registration and segmentation and making it a little easier. So the first application, as mentioned briefly earlier, is the MR and ultrasound registration during neurosurgery procedures, particularly the resection of brain tumors. And so we're using these, Sandy's team are using these two technologies, the variational autoencoders and the hierarchical models, and this has resulted in a MICAI 2023 submission. And just a couple of images to try to explain this concept. You can see on the left a brain ultrasound image, and on the right a brain MRI image. And looking at the various pixels in this space, excuse me, I went backwards, didn't I? There we go. You can see that there is intensity distributions across the pixel level, and the goal is to try to be able to capture that content and allow that to be analyzed further for registration purposes. And this is the image segmentation, and then ultimately the resulting registration on the right, showing you the results of this application. And so it allows us to add robustness to our methodology. The data that's being used here is being acquired in the Amigo facility, which I didn't have time to explain a lot about, but this is the Advanced Multimodal Image Guided Operating Room, a large translational research space that we have here at the Brigham, where we actually perform a lot of these procedures in humans and clinical trials. So we've done the data set that Sandy's working on consists of 66 patients to date, and you can read the inclusion and exclusion criteria here, and the pre-processing is done using the MRI images. And you can see the results here in this video, which I believe is playing, yep, before and after registration. So significant improvements being made in this area. Another separate project that Sandy and the team in collaboration with TRD1 and Stefan Meyer is working on Bayesian estimation of diffusivity spectra in both prostate and brain MRI images. And this is a collaboration also with CF Weston's P41 and Ron's in the NAC, where the problem really is to observe diffusion MRI signal decay. And that is the problem. And then we want to try to predict which fractions are decaying fast and which are decaying slowly. And so they're being calculated using Laplace transformations. There are many difficulties associated, as you can see here, and I want to show you some of the results that were presented last year at ISMRM, where you can see the difference in diffusivity values for normal prostate tissue versus tumor tissue. And you can see, I hope, that there's a significantly higher level of slow diffusion coefficients, components in the tumor tissue versus the normal tissue, which is on the left here. On the left, you can see that there's a hint of perhaps two peaks in both slow and intermediate values, but clearly we need more data, and this is an ongoing project continuing both in prostate and in the brain. The last aim of TR&D2 is the infrastructure led by, for curating imaging data for deep learning, led by Andrei Fedorov in collaboration with Nadia, and an external collaboration which is a new collaboration for us with Marco Nolden at DKFZ in Heidelberg, Germany. And the goals of this team are to develop a platform to manage and support data interaction, to integrate the analytic tools and the development of use cases with the data/platform, development of the DICOM standard, and harmonization of the data into a common format and nomenclature. So this goes to a lot of the same issues that David Kennedy was speaking about earlier. And one of the exciting new developments is that we have been exploring the opportunity to utilize the Kapana platform, which is a software platform available for medical image curation and analysis. I've highlighted in yellow some of the features that I think are particularly exciting about Kapana. Andre had provided all of them, but there's many features of Kapana that are very, very exciting, and particularly that will integrate with your local clinical systems. You can use it obviously to visualize, annotate, cohort build, but also to deploy and evaluate tools. Its data formatting can be with the DICOM and use it for segmentations and reporting. And it has great growth potential for interoperability, scalability, and flexibility. And the approach that Andre and his team are taking is outlined here, using 3D Slicer, using streamlined annotations from 3D Slicer. The data is integrated and harmonized across. And the use cases being, for the moment, our prostate biopsy data sets, which we have a large number of, which I'll show in a minute. But this illustrates a screen save from one of the visualization dashboards, and you can see some of the numbers of patients, the breakdown of many of the variables that are so important to understand when we're looking at data, be it the number of studies, the number of series, the field strength, and many, many, many other variables that can be occurring. And one of the important integral parts of all of this, of course, is data curation and annotation as well, of course. So again, based on our prostate biopsy program, which is led by my colleague, Dr. Kamal Tonkali, and the team here, we've been able to look at approximately 700 biopsy cases where we've been imaging in the bore before, during, and after a biopsy with a large amount of data, lots of segmentation, and also, of course, a lot of the correlative reporting that goes with these cases, be it the actual radiology report, the biopsy report, and the pathology report. And you can see laid out here just a screenshot of 3 of those groups of variables and some of the dense data that belongs in each of these, and how important it is to try to correlate across the system, that we have all of this data available to perform these analyses. So moving now to TR&D3, the last of the 3 TR&Ds. This is the Enabling Technologies group led by Nobi Hadda here at the Brigham. There are 3 aims and 3 teams in this TR&D. There's Dr. Hadda's team, Dr. Jagadisan, and Dr. Jonas, and you can see the aims lined up here. I'm going to go through them, so we will come back, we'll follow through on this. But the first one is needle guidance, and this is where Nobi is working with myself and others in prostate biopsy again, and Dr. Tonkali, to really develop techniques for accurately targeting not just a lesion, which may be as big as a centimeter, but subregions within that. Because as we know from TR&D1 and lots of other data, there's a lot of heterogeneity within given tumors. So we would like to be able to correlate pathologically these areas of heterogeneity. So precise needle placement is needed for that. And so we've been working for some time now on the templated biopsy approach and looking at the needles and looking at the data to determine what really changes and what can cause deflection and how much deflection of a needle will occur on insertion as it passes through various tissue planes to reach the tip and to really sample subregions of the lesion. So we've been recording case data using different gauge needles, different sizes, looking at the bevel tip approach, which is used to orient needles, and then registering those intra-procedurally. Nobi is collaborating with a longstanding friend and colleague at Worcester Polytechnic, Greg Fisher. Greg has, and Nobi and many of others of us are collaborating through a new NIH R01 to develop and validate cooperatively controlled hands-on robotic needle placement manipulators to really allow for this fine-tuned placement. And so you can see how the deflection work would play into that straight away. See if I can advance. There we go. So looking at the robot, it's all MRI compatible. This is actually in the Amigo MR scanner. Everything has been designed to be MR compatible to allow for full function, of course, and guidance. And then this little video will give you some concept of how this would be displayed during the insertion of a needle and in the teal color and the target point identified in a phantom here, using the 3D slicer and OpenIGT link to correlate in 3-dimensional space what's happening and how the needle is moving towards the target. And is there any deflection? Is there any alteration of navigation? And so that is the work they're doing in that area. So in TR&D3, AIM2, led by Jay Jagadisan, he's been working on trackerless navigation systems for many different organs. And right now he's focused on the skull-based surgery. Skull-based surgery is a complicated area to operate because there's a lot of dense bone in our skull bases and soft tissue. And it really can be hard for the surgeon to know how far in to go and how far to cut before they might lead to complications, as is illustrated here. If you use a microscope to assist in the retrosigmoid craniotomy for tumor resection, one of the complications that can happen is you break through the bone and cause a resultant CSF leak. And in this particular example, a patient had a CSF rhinorrhea where CSF fluid was leaking through the nose. This is something that you clearly want to avoid because that then becomes a source and potential route of significant infection. So the ability to three-dimensionally reconstruct images with bone overlays and soft tissue segmented behind the bone to demonstrate the tumor or the areas that want to be resected or not resected is critically important. And Jay has used a modified SLAM-based surface reconstruction approach, which is to add robustness to the surgical procedure. And I'm not going to walk through all of this because, to be honest, I'm not terribly sure I know exactly what's happening at every step here, but essentially it's integrated with the endoscopic motion and allows for a three-dimensional dense point cloud as the output. And I'll try to show you that now in picture form, where we start here with the original endoscopic video showing what the surgeon can see on the skull and at the base of the skull before the resection of the ear flap and the removal of the tumor. And then you can see an augmented video here now within yellow, the structures that are critical to be aware of and to resect. And so integrating all of that together is an extremely important advance for this type of surgery. In the last aim of this group, it's led by Oliver Jonas in collaboration with Sharath Bagalatova, one of my colleagues in radiology, one of the MDs here. Oliver has for a long time now been working on micro devices that can be inserted. These are tiny little micro devices about the size of a grain of rice that can be placed inside tumors. And before I go into that in further detail, these little devices have tiny wells within them that contain chemotherapy. And when you place the device into a tumor, the chemotherapy, of course, diffuses outwards into the tissue and allows for a local reaction in the tissue to determine responsiveness or non-responsiveness to that specific chemotherapy agent. So one of the trials that he's working on currently is in glioblastoma multiforme, where they have just completed an intrasurgical pilot study in 6 patients. And this was the insertion of the device at the beginning of the procedure, and then the resection of the tumor and removal of the device on the same day on the same patient, of course. And they're extending this because I'll present some of this data in a minute, but the next step will be to do a longer study where the device will be placed in several days before the surgical resection. And this is being done in collaboration through one of our CPs with the team at UCSF. The next step is, well, of course, to not have to surgically fully resect the tumor, but to be able to actually retrieve these devices in smaller tumors and in places when it's placed in percutaneously. And that work's being done and led by Sharath. And just to show an overview of what's happening in this in these patients where the device was inserted at the beginning of the procedure and then removed, you can see here the area of the tumor, and then a small device is placed inside it. Surgical resection of the tumor itself occurs, and this is the resulting specimen, excuse me, which is then obviously analyzed pathologically. But first thing, of course, in any type of procedure like this is to assess safety and feasibility. And it was feasible in all 6 cases that the devices were retrieved in 100% of them. And you can see that they were usable in almost all. One of them, the tissue was not accessible. So one specimen was not usable, but all very successful with no complications either early in the first 2 days or in less than 30 days. So it appears to be relatively safe, extremely safe. And you can see the impact, a slight lengthening of the surgery, no change in the overall cost, of course, because this is covered by the research expense. And then the ICU stay slightly larger maybe, and total hospital stay, interestingly, was less. And then the value of this, the value is that, of course, you take the device then and look at the tissue surrounding the device to see what, if anything, is happening to the tissue in response to the various chemotherapy agents. And temozolomide is one of the key chemotherapy agents used for the treatment of patients with glioblastoma. And it's all plotted out here. And you can see in one patient, there was a very significant tumor response. In 2 patients, there was no response in the adjacent tissue. And does that play out eventually? Does that mean that we can go forward in this patient with temozolomide, but not in the other 2? Of course, it's far too early to say yet. But a lot of this will be very, very valuable data to be able to be assessed in the future. And the new study is going to look at this 3-day dwell, and you can see a device being inserted here in this patient who has partial seizures from his tumor. And there's the size of the little tiny burr hole that's been created essentially here to allow the device to be placed in the tumor. And then there's a retaining catheter on the outside, which will allow for retrieval later on. So that's the end of that presentation of all of our science. I want to finish up with the area of training and dissemination and share with you what Tina and Mariana have been assisting us with. And like many others, our group is also doing serious numbers of lectures around the country, some both in person. And as CF mentioned, of course, we're adapting in a very big way to doing a lot of these things on Zoom and capturing and video recording these and saving them online for folks to come and avail of at any time after lectures are given is so very important. And so this is just an example of some of those. Project Week is also a nice overlapping collaborative effort between the NAC team and the RP41, the at NCIGT group. And here you can see similar illustrations to the report that CF just gave you on the most recent Project Week meeting, which I believe was the very first ever hybrid one, where it was both in person and online. And of course, this led to very significant increase in numbers of attendees, and the reach of this is really fantastic now. We've also been doing separate workshops, and every year, typically, we're hosting an image-guided therapy workshop. And thanks to my colleague Nobi Hatta, who hosted this one last year, we had, again, this was 100% virtual last year, and you can see the numbers here, significant numbers of keynote speakers. We had nearly 300 registered attendees from all walks of life in NIH and industry and academia, plenary talks, orals, short orals, and posters. A very exciting event. We, of course, like everybody else, have been attempting to train and educate through this P41. We've had 52 trainees since the beginning of this new P41 in 2021. And we currently have 29 trainees with us at the moment. Our CPs and SPs are here illustrated for you across the country. We've made some changes that we will be getting approval of later this year. I'm just going to try and highlight those for you. Already mentioned was Marco Nolden, added in as a collaborative project here for TR&D2, working with Andrei Fedorov. And Natalie continues to work with Jan Zakaria, but in a different grant mechanism. Now this has changed slightly. And then we've also added a new collaboration here for Natalie, working with Daniel Wall at the University of Michigan. So I wanted to take this time to thank everybody for all of their help in putting together this extensive presentation. I hope I didn't go over, and thank you very much for your attention.
Yeah, thank you everyone for the wonderful presentation. And now we move to a Q&A session. Let me start off with a general question to all the P41 centers. And you kind of indicated in the presentation, but my question is that if someone wants to start a collaboration with you, either as a CP or SP or as a new potential project, what's the process? Let's go reverse order. So I have Dr. Tempany right here, and maybe start with you.
Well, I'm going to take a quick stab at this, but I see I invited a lot of my colleagues to be online, and Tina's online, so maybe she can help me with this. But essentially, it's direct contact. We have, of course, an active website, which has just recently undergone a complete overhaul, and there are online forums there that people can complete. And of course, it's all through word of mouth as well, direct contact with colleagues at meetings and other ways. And Tina, would you like to, is there anything else to add to that, please?
I think direct contact is the main one in the IGT workshop that we've been running. We work with all the program officers of grants, not just from NIBIB, but also NCI and other institutes. Anybody who's working in our space of research, we look to see who are the latest funded grants, and we reach out to people to make connection, not just with us, Chi, but also with others who are in the field. So that's the main.
Yeah, thank you, Dr. Westy.
So I think we have outlined on our website a little bit the procedure, how to connect new project. But since we're sunsetting now, I think the best thing would be to connect directly with me or Ronki Kines and ask, because we have to a little bit figure out how to go forward with this kind of effort. And we're trying to, I mean, we're also inviting the NIH people to kind of tell us what could be a next center around, especially around our new magnet. But so let's make it more informal now. Send the questions to us directly, email to me or Ronki Kines, and then we can think about how we can collaborate.
Dr. Kennedy?
Yep, very similar answer. Claire's answer is contact us directly from our website. You see who our current ones are, and there's an invitation to contact us, and our access contacts are there, and we're happy to talk about ways to work that out and prioritize, people's interests and needs. In order to accomplish, things, we try to pool people together so that solving one problem, helps multiple people. So we look to how to package our collaborator and service requests to help manage the effort.
And Dr. Toga?
Yeah, no, I think it's the same for us. We're probably more in CF's world that, as we sunset, it'll become less formalized, certainly. But, we're always looking for good collaborations, and so reaching out to us, we're easy to find.
And in the Q&A, there are a few questions specific for Dr. Kennedy. I will let Andrew to ask those questions.
Thanks, Chi. Yes, so the first question for you, Dr. Kennedy, basically says, reproducibility is hard. It seems like it would take significant effort to implement your strategies. So what are the largest roadblocks to implementing them? And is there some learning curve that becomes less burdensome with experience?
Yes, it's hard. And my first answer is you don't have to do it all at once. There's a whole bunch of different things which have variability in terms of their hardness. So in general, we encourage people to pick off bits and pieces that they can improve on, in sort of each subsequent, paper, and try to overcome, different things, individually. Getting center support, so you're not doing it on your own. Make sure your, supervisors, are behind your effort and supporting you as well. And I should advertise, it's referenced briefly in the talk, but a paper by Keith Bush, who took a center at the University of Arkansas, and during pandemic, sort of went whole hog, into trying to solve some of these issues. And it's costly, and it takes sort of top down desire to do this if you're trying to do everything all at once. So he has some great learning things there. But ultimately, we're here to help, and we can help you identify pieces that can be accomplished and sort of move slowly towards this long-term objective.
Thank you. I'm going to throw another one or two more your way. So this one's regarding annotation, which you talked extensively about. Obviously, it's very important for reproducibility. How do you balance between annotating kind of the data too much and ending up with a huge number of annotations that is not practical to keep track of, which suggests using as few annotation terms as possible?
Yeah, so that's an important balance. Good computer management of annotations, I'm not sure we'd ever have a problem with too many annotations. Then it's just a matter of what the interface to dealing with them is. So for example, our electronic DICOMs and data sources have lots of annotations aligned with them from the DICOM sources. And so there's ways to keep that attached to the data all along. The much more challenging place where it really gets to the end user is the behavioral and clinical and assessments that happen. You could make up all sorts of things about a mental health exam or a various behavioral assessments, and you don't want to go too crazy annotating all that. You want to be clear about what test was done, what summary metric this is, so that can be traced. The community can argue further about what the exact sort of behavioral meaning of an MoCA score of 27 actually means. That can evolve. but making sure that, this is a MoCA measurement, it's a particular assessment, and sort of the low level sort of minimum description of what was done, needs to be annotated so that, that sort of reasoning about, its influence and its uses, can evolve over time. So yes, don't go too crazy, but make sure, the place that measurement, falls in terms of what measurement, what, domains it's, accessing can be done, with sort of a minimal metadata, and the rest can evolve over time.
Yeah, thank you. This is Chi again. Considering the time, I would like to pass it back to Beirut. And I do see we have remaining questions, and Tina already invited Dr. Kennedy to have a further discussion, and we're glad to see some further collaboration between P41 centers. And now pass back to you, Beirut.
Thank you, everyone. It was great. Really, I really enjoyed it, and thank you for all your time and effort. Well, I followed the question that is going on is how do you guys, with what you learn? I know that you know about each other's works, but how do you see a collaboration based on what you just learned? Is anything comes to your mind that you want to throw it out? Anything on the training or even the tools that you can utilize from each other? So some of the things I'd like to see that is basically, if you're working in the same disease or in the same area and you have a software or tool that one develop, and to be able to collaborate by just utilizing those things. So not collaborate on developing, is basically to be able to be each other's users, service projects. Or even, to combine it to different tools.
Well, I think, Baruz, the issue for us, certainly probably for CF too, is, the clock is ticking. So, we have some months left. we can get done what we can get done, but, once the funding stops, those efforts have to be redirected to other directions. So some of these things actually take a bit of time to try to find an area of alignment and ways in which you can work together in a productive way. So, we're all friends. That's a good thing. And I think, you're going to see in the future, we're all going to end up working together in some fashion or another anyway, only because we enjoy each other's science and company for that matter. So, I think we're going to have to reconfigure some things just based on necessity.
And you don't have to do it now. By the way, that is, I really, sometime we should talk. I'd be interested to know what is your plan to do with it, because I don't want everything that you have done to be just disappear. So is it that, are you planning for any, the spin out?
Yeah, it's absolutely not going to disappear. We have some cemented, and my guess is that's true for all of us. The efforts that are undertaken as part of the resource end up establishing research directions and programs that stand the test of time. They just do. And so, nobody's going to walk away from these things. We just have to figure out how to keep them in the most productive state that they can be in. It's not just a responsibility. It's being responsive to the people that you're working with. So we have to keep doing this. The mechanism yet, not sure. But we will keep doing this.
Yeah,
I agree with that. And so we're all friends, and we are working together in different ways. But if we do it in the same P41 manner or not, I mean, we might, but we will continue to work in some way. So, and I learned a lot, by the other centers. So yes, I have some ideas.
Great. I mean, the idea can come later, but it's we should talk sometimes. I try to push PO1 back to the NIBIB, but because of the budget, I stopped doing it, because if I do that, because we don't have a budget, then Bruce may be agreeing, but it's going to cut my P41 to go to that, and I don't want that to happen. So it's a tough call. So, but hopefully other ICs can do that.
Yeah, so I'd like to be able to reach out with you, but I'm sure CF would like to as well. And, we can just brainstorm and see what's possible here. Not stepping on anyone else's toes, but seeing if there's mechanisms that make some sense for everyone. So.
Great. Good day. Any other question from any one of you? Okay, I'd like to thank all of you. I appreciate everyone. I acknowledge everyone's work. I had a slide with the pictures, but not everybody's pictures. So since you are all I'm going to appreciate all, but I'm going to also acknowledge Asha's work that he has made this happen. So as the Ryan's great work, because it's the second seminar, we had just gone smooth and we had no issue before, and we didn't have it here. Thank you, Ryan. And then at the same time, thanks to Chi and Andrew. So it was great seeing you all, and keep in touch with me and with each other. So if you come up with any idea, I'm all here.
Sounds good.
Okay,
Very nice to see you all. Okay.
Thank you for hosting us.
Thank you.
Thank you.
Yeah, thank you.
everyone.
See you,
Bye,
everyone.
everybody.
Coming.
Bye.
Bye.
Be well.
Thank you.